Hey guys, so today's session will be based on bacterial and phage genetics. Bear with me as we go through this because this is my first time doing an online lecture. So, thinking about bacterial genetics and phage genetics, what the first thing we should do is understand what we know about bacteria so far. We know that it is a prokaryote with no membrane-bound organelles. The prokaryotes are haploid and they contain a single circular chromosome. So here you can see the single chromosomal DNA called the nucleoid. It lies in the cytoplasm. In the cytoplasm itself, you also have small circular DNAs called plasmids, and these are actually independent DNA molecules from the chromosomal DNA, right? Since bacteria, as we know, bacteria are haploid. There are no diploid versions of them like fungi. Fungi can change between in diploid and haploid depending on the circumstance. But in the case of bacteria, they do not actually change into they do not change into a diploid cell. And in this case, they do not undergo meiosis. So in this case, replication begins at a fixed point, but it is bidirectional. We will talk more about how replication works in the next session. So one of the first things we talk about is how does this reproduce, right? So how does a bacteria reproduce? It is through a process called binary fission. Essentially, what you guys need to know is that it is a version of asexual reproduction, right? Dr. Bruce didn't go into specifics of how binary fission works, but this is more to give you guys an idea as to how that would occur. So the first step is replicating DNA. You replicate the DNA, you go in there, it separates out a little bit to like it pulls the membrane a little bit farther away, and then it starts cytokinesis. Do you guys remember what cytokinesis was? Does the division of the cell, right? The cell itself. So the cell membrane kind of pushes in, it slowly kind of becomes that wall, and then it separates out into two daughter cells. So essentially it's taking out the nuclear division because there is no nucleus in a prokaryote, right? So essentially what you guys need to know about bacterial reproduction is that it is an asexual process. Okay? So the next thing about bacteria is that it is a convenient way to understand and study different genetic analyses. So one of the main reasons why it is a good organism is that it is easy to grow and maintain. And to actually grow a bacterial culture, we have two methods. Do you guys remember what those were? So those would be liquid culture, where you use a liquid of different salts and a little bit of sugar just to get these bacteria growing. And then it's a similar thing with solid medium plating, like agar plates and so on and so forth with those guys. So thinking of solid medias and plates and all that, what exactly is in the media that is used for solid plating when you're studying bacterial genetics? So the medium that we use is called minimal medium. What is in it? So minimal medium is essentially a medium that contains salts, a carbon source for energy, and water. The minimal medium itself does not require a special supplement, right? So in most cases, a solid plate would look like this guy, like this, these plates that are shown here. So what are the differences between autotrophs, auxotrophs, and prototrophs, right? So prototrophs and auxotrophs are two categories of bacteria. And this includes prototrophs, which are our wild type bacteria. It can grow in the minimal media itself without requiring any special supplements. Whereas auxotrophs, they're mutants and that cannot grow on minimal media with without special requirements. So when you have an auxotroph, you have something supplemented to the media to allow them to grow. Okay? So to see this, to see and designate these different prototrophs and auxotrophs, you use different genetic symbols. And so for an example, they say bio-negative. So we have the symbol bio-negative. This shows that it requires biotin added as a supplement to the minimal media. So bio-negative means that that specific strain of bacteria, that auxotrophic bacteria, requires the strain itself lacks biotin, and for for that strain to grow, you need that you need biotin as a supplement to that medium. This is the same thing going on with all of the all all of these amino acids. Sorry about that. So you need, so when you have a negative next to like arg, which is standing for arginine, one of the amino acids, it's saying that it lacks arginine and it's needed as a supplement to the minimal media. Same thing here. It's like methionine. It adds on there. You need that as a supplement. So any amino acids or kind of like probiotic, that kind of things, if you have a negative with it, that means it's typically required as a supplement for it to grow. If you have like lac or gal, which are like lactose and galactose and different other different sugars, then you cannot, if those guys have a negative next to it, you cannot use that as the sugar or carbon source in the minimal media. You have to use something else to allow those strains to grow. And then with antibiotics, you have, so STR is streptomycin, and the superscript R refers refers to resistance. The superscript S is for sensitive. So if it is STRS, you know that it's sensitive to the antibiotic streptomycin. Whereas if you have R as a superscript with SGR superscript R, you know that it is resistant to streptomycin, right? So these are some examples of how you would use those guys. There are many more, but in this, we're just going to focus on these guys. So just to get a hang of, so here's an example. If strain A would only grow if the medium were supplemented with methionine, and strain B would only grow if it were supplemented with threonine, leucine, and thymine, how would one designate these strains? So where do we start? We start off asking, what do we know about strain A? In this case, we know that with strain A, it only grows when supplemented with methionine and biotin. So this tells us that strain A is deficient or lacking methionine and biotin. And it requires a minimal media that is supplemented by methionine and biotin. So this gives us the designation for strain A, met negative, bio negative, and then everything else positive, because it is lacking methionine and biotin. That's why we keep them negative in this case. So what do we know about strain B in this scenario? We know that it only grows when it is supplemented with threonine, leucine, and thymine. This means that it is deficient or lacking any of these three guys. And that if you want this strain to grow, you need something that is supplementing these three guys. So with that, you can say that you can designate strain B as met positive, bio positive, threonine negative, leucine negative, thymine negative. Okay, let's do another example. If strain A would only grow if the medium were supplemented with threonine and biotin, and strain B would only grow if it were supplemented with methionine and thymine, how would one designate the strains? So what do we know? We know that strain A only grows in supplemented, only when supplemented with threonine and biotin. This means that this strain is actually lacking those two. So we get this designation here: met positive, bio negative, threonine negative, leucine positive, and thymine positive. So essentially, it's whichever one you're supplementing with, that's the ones that ends up with the negative. So with strain B, you know that it needs methionine and thymine, which shows that it's lacking it. So you end up getting this as an answer for how to designate strain B. So everything except methionine and thymine are positive. One more question of this guy. If strain A would only grow if the medium were supplemented with methionine, biotin, and leucine, and strain B would only grow if it were supplemented with threonine and thymine, how would one designate these strains? So what do we know? We know that this only grows in methionine, biotin, and leucine. We see that, so now we know they are lacking it and it requires that supplement. So this is how we get this guy. So it is negative for methionine, biotin, and leucine because that's what they are lacking and that is what it is showing here in this question. So now what do we know about our strain B in this case? We know that it only grows when supplemented with threonine and thiamine. This essentially, now we know that we can get this guy. So this is how you would designate this strain because so since strain B is lacking threonine and thiamine, this is the strain that this is how you would designate strain B in this case. Okay, so we talked about different types of auxotrophs and all that, but then we never went into the fact of what happens when these auxotrophs mix with each other, right? So so to understand this further, they actually took strain A, which is this guy, with methionine and biotin required as a supplement on the plate. So we have these guys, we just have strain A just sitting there, the barely couple of cells that we have. Then we have this guy where we have strain B, which is plated. We see nothing happening, right? Just sitting there, nothing fun, nothing exciting. But then when you mix them, you see a mix of strain A, strain B, and then we get prototrophic wild type colonies. Like, what? So these guys actually show up when all of them are positive. They grow into mini colonies like, hey, hi there. How's it going? What's happening? Right? So when these guys came in, they're like, ah, so recombination must be occurring, right? But then we had other scientists argue it could just be the cells leaking and the bacteria just picking up different components to make it actually turn into prototrophic colonies. So scientists were like, let's try this method. So they put a strain here in this side of the horseshoe, plugged it up here, and then we put B strain here, separated them with a filter, kept a suction going, and the only thing that could pass through these filters were anything liquid. No cells, no organelles, nothing. Sorry about that. So, but no cells could move around in there. And after a little while, they incubated it, they plated it, and they saw strain A, strain B, but they had no prototrophic colonies. So this is how we kind of led to what is the basics of bacterial genetic exchange. So we know that recombination allows DNA to generate new allelic combinations. This combines by this combination of alleles are actually generated through the transfer of DNA from one cell to another, and this transfer of DNA occurs in one direction and not the band, I promise. It's only in one direction, unidirectional, between the donor and a recipient. And then you have only a partial transfer. So not the entire chromosome is moving into the recipient. So what are the three mechanisms of DNA transfer or bacterial sexual processes? So three mechanisms of DNA transfer include transformation, transduction, and conjugation. So what is transformation? Transformation is when a bacterial cell takes up a free piece of DNA lying around in its surrounding. So it literally just pulls that right in and incorporates it into the chromosomal DNA. Now we go into transduction. Transduction is when gene transfer occurs from a donor to a recipient by the means of a bacteriophage. Now you might be thinking, what is a bacteriophage? So bacteriophage is a virus that infects bacteria. So phage is a virus, bacterio is just a fancy way to say bacteria. And in this case, bacteriophage is essentially a virus that infects bacteria. And in these cases, when they start infections, they can either be in the lytic cycle, which is essentially they multiply it and then they multiply in the cell and then lyse them, so killing them immediately. Whereas with lysogenic cycles, it can either go directly into lytic cycle or it stays dormant and until an external stimuli comes in and be like, oh, time to go into lytic cycle, poo, and go boom, right? So essentially with transduction, you do not need physical contact between the cell, the donor cell and the receiving cell. It also is DNA resistant, which means it it is resistant to any kind of breakdown, right? So next method would be conjugation. This is the one that Dr. Bruce ends up going more in depth with in your slides. So with conjugation, you have a process where a bacterium transfers genetic material through direct contact. So it's essentially touching each other. And this ability to come together as conjugates depends on this plasmid called the F plasmid, or the fertility factor plasmid, okay? So how does conjugation actually work? So the F plasmid, if a cell has the F plasmid, it's called the F positive cell or donor cell. So this guy goes, and then, so this guy here, we have an F plasmid, it's the donor. Then we have a recipient, which doesn't have the F plasmid. So it cannot do anything right now. And it's called the F negative or the recipient cell. So the next thing would be is that so the F positive ones have something called a pilus, or in a pilus. And this pilus comes and attaches itself to the recipient cell, and this pili actually lets it come together, and it transfers that DNA into the F plasmid into it, and then it becomes a new donor. So that DNA, the F plasmid ends up getting transferred in, and now they both become F positive ones or donor cells. Hope this makes sense so far. Okay, so what are high frequency recombination HFR strains? So HFR strains are actually a derivative of these F plasmids. And with the ability to so this specific derivative has the ability to promote high frequency of recombination. And these strains have the fertility factor inserted at different points, and different directions, depending on how it ends up coming in, attaching to the chromosome itself. So when it gets attached to the chromosome itself, it this this integration ends up determining the linear order of the transfer markers for each HFR strain. So for example here, so this is the fertility factor that we have. The origin would be the arrowhead, and this red rectangle would be our last thing to enter. So when we have this guy, we would go on by saying threonine, proline, lactose, and so on and so forth. It kind of goes in that direction. Whereas with this guy, we have threonine, thymine, glycine, histidine, galactose, so on and so forth in that direction, just because the the orientation itself is different for each of them, even though it's the same sequence itself. So what are two main features of the HFR strain? The HFR strain allows the production of recombinants 100 times more than the normal F strain, and faster. And if there is no recombination, the fragments end up lost when cell division occurs. So if you have virtually in these cases where you have HFR and F negative, where these haven't actually recombined, then virtually none of these F minus parents end up converted to F positive or into HFR strains. So scientists started to investigate how are the HFR genes transmitted to F negative cells during a cross. So they tried to study this with two with 2 bacterial strains, but two these two genotypes, they mixed them together, and after specific times they took some samples, they took it, they put it into a blender, shook it, like they blended it, and that procedure is to separate out these donor and recipient cells that were mating to like transfer DNA with each other, right? So when you take them out each couple of minutes or so, you end up using a procedure called interrupted mating. So with this interrupted mating technique, they found out that each donor allele first appears in the F negative recipients at a specific time after mating begins. So azithromycin here came out at 10. This guy came out around 11ish. This was around 17. Like, it comes out at a specific time. Certain markers come out certain times. And they appear in a specific sequence. And later markers or alleles end up very few in frequency. So if you look at this, you have this many of this guy, so, and it's in the beginning, whereas with galactose, we have it here very low frequency, around 25, 30-ish. So it depends. So when it comes in, you know that if it's in the first in line, it comes in first and it's more frequent, whereas the later alleles end up not being as frequent in the recipient cells. So in this case, it always starts off from a fixed point. Whenever it goes in with a fixed point, which is termed origin. So the origin goes in first and then everything else follows. So now the origin is known as the site where the plasmid is inserted, right? So the farther a gene is from, so we have the origin. If we have a gene that is farther away, it gets in later on. So we have this guy entering in at 10 minutes, whereas this guy took around 25 minutes to enter that cell. And it typically stops before the final ones can even get in there. So the later genes end up not getting included in the X conjugates. So using this method, you can actually determine the order of the genes by actually keeping track of what time they came in. So when it comes in, you can see which order they're coming in, and that would be the order that they are mapped on a chromosome. So you can make a chromosome map based on the number of minutes or unit of minutes. I will go over another example, an example of this later on. There is one example in the question itself and Dr. Bruce's slides. So try the study guide out. This is This slides are not as comprehensive as the slides that Dr. Bruce gives or the study guide itself. So make sure to look at those questions, try out the questions in the study guide itself, and let me know if you guys have any questions. And let's get through this one more question and we'll be done. What are two forms of recombination in bacteria? So lytic and lysogenic, they're not recombining in bacteria. That's more bacteriophages, how they interact with the bacteria itself. Auxotrophic and prototrophic are actually, they can recombine with each other, but the thing is, those guys are classes of bacteria. So one is the wild type and one is a mutant. So those are categories of these bacteria. Then we have insertion and replication. Those are not recombining anything. That is not recombination whatsoever. Then we're down to these two choices. So we have mixed and conjugation. Mixed is not exactly a form of recombination in bacteria. We have three types of DNA transfer moments, right? So in this case, the answer here is conjugation and transduction, because those two are the main options that we have, right? So this is what I have for now. Let me know if you guys have any questions or concerns. Make sure to look into the study guide and try practicing the questions from the study guide with Dr. Bruce's slides. Look through the questions that he explains, because those are mostly the questions that he will be using as exam questions, possibly. I am not entirely sure, but make sure you know those kind of questions and try to attempt them. So, and then with the harder questions on the study guide, you guys can come to me, talk to me through email or Zoom. Just let me know if you guys have any questions, concerns. Stay safe and healthy, guys, okay? Don't want any of you guys sick. So, until next time.